All right, BIMM 100 students, let's get you ready for that final exam. We're going to do a deep dive into lectures 4 and 5. This is the really cool stuff about how our cells copy and protect their genetic blueprint. Super important stuff. Okay, so here's our roadmap. We're going to cover the replication playbook, that two-lane highway problem, how we solve the issue at the very end of our DNA. We'll meet the different polymerases, and then wrap up with how to actually analyze the message. Let's jump in.
All right, first up, the replication playbook. Think of this as assembling the all-star team. These are the enzymes, the little molecular machines that have to get in position right at the starting line to kick this whole thing off. So let's just pause and think about this for a sec. How does a cell flawlessly copy over 3 times billion base pairs of DNA before it divides? I mean, that is a mind-boggling number. It's a huge challenge, and the cell pulls it off with this incredible, perfectly choreographed dance of enzymes. First on the scene, we've got helicase. You can call it the unzipper. Its job is simple, but absolutely critical. It just plows down the DNA, acting like a zipper on your jacket, prying apart those 2 strands to expose the code inside so we can actually copy it. Now, as helicase is unzipping everything, it creates a ton of tension up ahead. Imagine trying to quickly unwind a tightly coiled rope. It just gets knotted up, right? Well, that's where topoisomerase comes in. It's the stress reliever. It works ahead of the fork, making these little nicks in the DNA to release that tension, preventing the whole thing from turning into a tangled mess. Okay, so here's a really important rule you gotta remember. DNA polymerase, our main builder enzyme, is a bit picky. It can't just start building on a bare strand of DNA. It needs a starting point, like a little welcome mat. That's primase's job. Primase is the initiator. It lays down a short little piece of RNA, called a primer, which gives DNA polymerase the perfect spot, that 3' OH group, to grab onto and start building. Okay, so the team is assembled, we're ready to go. But we hit a major snag. The 2 strands of DNA run in opposite directions. It's like a 2-lane highway where the cars are going opposite ways. You can't just use one simple strategy to copy both at the same time. You need a clever workaround. So this opposite direction thing creates a really interesting puzzle. The leading strand, that one's easy. It's like driving down the highway in the same direction as traffic. It's synthesized continuously in one long, smooth piece, just following right behind the helicase. But the lagging strand, that one's going the wrong way. It has to be built discontinuously, kind of backwards, in these little chunks. It's a much more complex process. So how does this work? It's like a perfectly choreographed 5-step process. First, primase comes in and lays down a bunch of RNA primers along the strand. Then, DNA polymerase comes in and builds the DNA in between them, creating these short fragments we call Okazaki fragments. Step 3, a cleanup crew, RNase H and Fen1 gets rid of those RNA primers. Step 4, DNA polymerase comes back to fill in the little gaps. And finally, DNA ligase seals everything up, like molecular glue, joining all those fragments into one continuous strand. Pretty cool, right? Okay, so that whole lagging strand synthesis thing is super clever, but it creates another problem. And this one happens right at the very tips of our chromosomes. It's called the end replication problem, and it's a huge deal. Here's the issue. When you get to the very, very end of the lagging strand, that last RNA primer gets removed, but there's nothing for DNA polymerase to grab onto to fill that last little gap. So what happens? With every single time a cell divides, the chromosome gets a tiny bit shorter. And you can imagine, if that keeps happening, you're eventually going to start losing really important genes. Not good. So how does the cell solve this? With a truly amazing enzyme called telomerase. It's the end extender. Its whole job is to add these repetitive DNA sequences, called telomeres, to the ends. The best analogy, and you should definitely remember this, is the little plastic tip on your shoelace. That's an aglet. It keeps the lace from unraveling. Telomeres do the exact same thing for our chromosomes. Now, this is a huge concept for your exam, so listen up. Telomerase is special. It's a reverse transcriptase. What does that mean? It makes DNA from an RNA template. And get this, it carries its own little RNA template around with it. This completely breaks the normal rules of the central dogma, which says DNA makes RNA makes protein. It's a really big deal. And the way it works is pretty straightforward. Telomerase latches onto the end of the chromosome. It uses its internal RNA template to add a new little chunk of DNA. Then it just scoots down and does it again and again. By extending the end like this, it creates enough of a buffer zone for primase to come in and lay down that final primer so the whole chromosome gets copied. This is absolutely critical for how long our cells can live and plays a massive role in things like aging and cancer.
Okay, let's talk about the polymerases. We've been saying DNA polymerase like it's just one thing, but in us eukaryotes, it's actually a whole family of different enzymes. And for the exam, you'll definitely want to know who does what and how good they are at their job.
So are they all equally accurate? No way, not even close. The cell is smart about this. It uses different polymerases for different jobs, and it only brings out the super accurate, high-fidelity ones when the DNA sequence is going to be permanent. It's all about efficiency while making sure the genome stays perfect.
So this table really lays it all out. You've got polymerase alpha, or Pol alpha. It works with primase just to get things started. Its fidelity is low, but who cares? That part gets removed anyway. But then you have the heavy hitters. Pol delta for the lagging strand, and Pol upsilon for the leading strand. Their work is the final product, so they have to be super accurate. They have very high fidelity.
All right, we've copied the DNA blueprint. Now what? The next step is to actually read the blueprint, right? That's transcription, where you make RNA. And as scientists, we need tools to see that RNA. One of the classic, fundamental techniques for doing this is called a northern blot.
A northern blot lets you answer a really key question, and definitely something you might see on the exam. It lets you ask, is my favorite gene actually turned on in these cells? You could compare, say, kidney cells to liver cells. Is the gene on in one but not the other? And you can also get an idea of how much RNA is being made. And the process for doing one is a classic lab procedure. First, you gotta crack open your cells, say kidney and liver, and isolate all the RNA. Then you separate all that RNA by size using a gel, kind of like a molecular race. Next, you blot, or transfer, that RNA onto a solid membrane to make it easier to work with. Then comes the magic part. You use a labeled probe, a piece of DNA that will only stick to the specific RNA you're looking for. Finally, you just detect where that probe stuck, and bam, you can see your RNA.
So, when you step back and look at the big picture, what's the takeaway here? It's that none of this is random. The cell has this incredibly precise, fine-tuned toolkit with all sorts of error checking built in. It's all about copying, protecting, and then reading those genetic instructions with the highest possible accuracy.
And that brings us to the final, really important question. We've just gone through this amazing, high-fidelity system. So what happens when it breaks? What happens when these systems fail? Well, that's when you get mutations, disease, and cancer. So knowing how it's supposed to work is the absolute key to understanding what happens when it goes wrong. Think about that as you're studying. Okay, that's it for this primer. Good luck on your exam.